So, hello everybody. This is yet another episode of the Western Biology Evolution Seminar Series. I'm your host, Matthias, and I'm here today with Professor Ben Evans. And I will let him introduce himself, and then he can jump right into his talk. Ben, thank you for being here with me today.
Hey, thank you, Matthias. So my name's Ben Evans. I'm a professor at McMaster University in the biology department. I'm interested in evolutionary genetics and genomics. And I've been studying primates, but also frogs. And I'm going to talk today about sex determination in frogs, and in particular, rapid evolution of genetic sex determination. And I wanted to begin by acknowledging some people, both at McMaster and at the National Xenopus Resource in the United States, and also beyond in various countries in Africa, where I have very productive collaborations with scientists there. And I also wanted to acknowledge that McMaster is on the traditional territories of the Anishinaabe and Haudenosaunee people, and it's protected by the Dish with One Spoon Wampum Agreement. So I wanted to, again, thank all of these people that are listed here. These are current and previous students and also collaborators that have really played important roles in this work, and also these funding agencies who have either directly or, in the case of NIH, indirectly supported me and the work I'll talk about today. And thank you again, Matthias, for the invitation. So one of the, I think, important and interesting aspects of biology is that when we look across species, oftentimes we'll find genes and genetic function that are broadly conserved across disparate organisms. And this is basically why we can use model organisms to understand human biology. And so, for example, the gene PAX6 is a gene that's involved with the development of eyes. And you can take the human PAX6 gene and ectopically or artificially express it in a fruit fly and cause an eye-like structure to develop in the wing of a fruit fly. Similarly, you can take the homologue of PAX6 from fruit flies, which is called eyeless, and express it in a frog, and you can cause the eye, an eye-like structure, to develop in a developing tadpole, and that's what's indicated on the bottom here. And so basically, the function of this gene has been conserved, for deep evolutionary time across diverse metazoans, as diverse as arthropods and humans. But that's not, of course, always the case. Sometimes what we see when we look across species is that there are phenotypes or characteristics that are similar across those species, but actually the genetic architecture, the genetic underpinnings of those traits actually is different, even though the phenotypes are the same. And so this is an example of exactly that. So if you look at the top of a fruit fly, either Drosophila melanogaster or Drosophila simulans, they actually look quite similar. If you look at the thorax, you'll see these bristles that basically look indistinguishable between these species. But when you make a hybrid of these two species, you can see that the bristle formation is aberrant, meaning that when you have the genes from each of these parental species put together in a hybrid individual, they don't work well together, and they cause abnormal bristle formation, even though the phenotypes in both parental species are actually indistinguishable. And so this is an example of a process that we call developmental systems drift, which is when you have two species that have similarities in some trait, but the genetic architecture that underpins that trait is actually different. And we see developmental systems drift operating throughout a variety of phenotypes across diverse organisms. And this is important because it influences the degree to which we can extrapolate findings from a model organism to ourselves. And so if, for example, a biological phenomenon is operating in a model organism, but there's been developmental systems drift between the divergence of that organism and humans, then that same process may not actually be relevant to humans. And so we see, again, there are a lot of examples of developmental systems drift. And one of the prime examples actually is in a fundamental feature of many sexual species, which is sexual differentiation. And so we know, for example, in therian mammals, so placental and marsupial mammals, that there's a well-characterized system for sex determination where a gene on the Y chromosome triggers the male differentiation pathway. And so, for example, you can take a mouse, and this has been done, that's a genetic female that has two X chromosomes. And if you add in the SRY gene, which is normally on the Y chromosome, and it's expresses and gets translated into a functional protein, you can sex reverse that genetic female into a phenotypic male. And that's what's shown here. On the right side is just a male individual that's XY, and then on the left side, and then on the right side is a female that's an XX individual that's expressed in an SRY gene, and she develops into a phenotypic male. And so that is sort of a well-characterized system. We think that it works like this, that a zygote is formed through fertilization. It develops into a multicellular organism that has what we call indifferent gonads that have the potential to either develop into a testis or an ovary. And if it's an XY individual and the SRY gene is expressed, then that causes this indifferent gonad to pursue the male phenotype and develop into testes and develop what we call Sertoli cells, which are a fundamental structure within the testes. And if the SRY gene is absent in an XX individual, then those indifferent gonads develop what we call granulosa cells, and that's part of the ovarian structure. And then we have a female phenotype. And obviously hormones are involved, and hormone receptors are involved downstream of these genes. But, there are also many other genes as well. So for example, in males, we know that DMRT1 is important for maintaining testicular structure and also promoting the development of Sertoli cells. And we know in females, the FOXL2 gene is involved with the development of granulosa cells. So there are diseases where one or the other gene has been incapacitated, and we see a consequence in terms of sexual differentiation where, for example, if in the absence of DMRT1, genetic male will be unable to develop testes and have the development of what we call a gonadal streak, so an undeveloped testis or undeveloped gonads in an adult individual. So that's our system that we think is operating across things as diverse as kangaroos and humans and mice, of course. But when we look at other organisms, we can see that there's still separate male and female individuals, but actually the genetic architecture that underpins sexual differentiation is quite different. So, for example, in birds, we have a completely different system for sex determination. There's still separate male and female individuals, but occasionally we can observe what are called gynandromorphs, which are depicted here. So this is an individual that is part male and part female. So this left side of the animal has male-specific coloration. It's got a wattle, and it's got a spur on its legs, which are all male phenotypes. And then the right side of the animal has female-specific coloration, no wattle and no spur. And so there's actually, a completely different system for developing for sexual differentiation, where we think that in birds, sexual differentiation is actually what we call cell autonomous. There's some characteristic of individual cells that allows that cell to develop into a male or a female phenotype. And so that's why one individual, in this case, it was probably a chimera between two genetic individuals that gave rise to this gynandromorph. But each individual cell in that two-cell stage had a sexual identity that was then promoted by the expression of genes such as DMRT1 and many other genes that also operate in humans and other species. And so this is an example, again, of developmental systems drift, where we have the same phenotype, separate male and female individuals, but a different genetic architecture that underpins sexual differentiation in these different organisms. And we can see this across, a broader array of diversity. I've already talked about theory in mammals, but another type of mammal, monotremes, like the platypus, actually lack an SRY gene altogether, and they have a completely different system for sex determination. There are examples, for example, in reptiles. Turtles have temperature-dependent sex determination, and even within that, there's sometimes a warmer temperature that makes you male, and sometimes a warmer temperature makes you female. And then fish, of course, have an incredible diversity of sex determining systems, including novel sex determining genes that have been discovered that are present in a few species and not present in other close relatives. And so there's really a lot of rapid evolution of the genetic basis and orchestration of sexual differentiation across the diversity of organisms that we have on our planet. And so one of the, I think, fascinating aspects about this diversity is that some components of sex determination systems often are broadly conserved, whereas others evolve rapidly. And so you have this sort of architecture, this machinery that develops that's involved with orchestrating sexual differentiation. And some components of that machine are similar across species, and then other components evolve really quickly. And so an example of a conserved component would be the DMRT1 gene, the double sex and MAB3 related transcription factor 1. So this gene was originally discovered in fruit flies and nematodes, and it was named after genes in homologs in each of those species. But it turns out DMRT1 is also involved, for example, in male differentiation in humans. And so this gene, we find it turning up not only as a downstream factor, but sometimes as a trigger for sex determination in many, many metazoans. At the same time, many other groups, not as much birds and mammals, which have generally stable sex determining triggers, but many other groups such as amphibians, for example, that I'll talk about today, have a lot of variation in the sort of light switch that turns on or off one or the other sexual differentiation pathway. So there's a lot of variation among different species of amphibians and also fruit flies and fish and reptiles in the genes that orchestrate sexual differentiation, and also the genomic locations of those genes. So which chromosome in the genome is a sex chromosome. And so I find this sort of a fascinating problem in biology. How do you have these sort of fast-moving, rapidly changing components interact with conserved components to give rise to this extraordinary variation that we see across species? And so we're trying to understand developmental systems drift of sex determination, and we're using as our model the frogs of the genus Xenopus, which are really an incredibly powerful system, as I hope to illustrate. So our goals are to first understand how sex determination is achieved by identifying and understanding the gene content in sex determining regions in the genome in different species. So we're taking a comparative genomics approach there. We're also trying to understand the processes that influence where new sex determining regions arise during developmental systems drift. And so we're specifically interested in looking at how what's called the recombination landscape influences the propensity of a particular genomic region to acquire a novel trigger for sex determination. And then finally, and this is what I'll end up with later on in the talk, we're using gene editing to try to tease apart and dissect out the different functional components of sex determining pathways with an aim of understanding how, at a functional level, these new sex determining systems actually arise in different species. And so the questions I want to focus on today include, first, a kind of survey across different species of Xenopus frog, which asks how much variation in sex chromosomes are there among species. So is there, for example, one system for sex determination, or are there many? I then want to look at what's called the recombination landscape and ask how that could potentially influence where new sex determining genes arise in the genome. And then finally, I want to talk about some of the results we have from genome editing that ask what do sex-related genes do now, and what did they do in the past, and how did they acquire their sex-determining and sex-related gene function. So first, let's ask about how much variation there is among species in African clawed frogs. And so to do this, we're basically using genomic approaches to map triggers for sex determination in different species. And this is done through a fairly straightforward approach. We're using reduced representation genome sequencing, and we're doing this across families that we've raised in the laboratory here at McMaster. And so the idea, again, is quite simple. Imagine that you have a mother and a father, and at a particular genomic position, there is a polymorphism. So there's a T in the mother, and then she also is heterozygous, so she has a C as well at that position. And then the father is a homozygous for a C residue in DNA. And imagine that we had offspring, and we could genotype those offspring as well. So imagine that that particular genomic position were autosomal. If that were the case, then the probability of a daughter or a son inheriting that T would be 50-50 from the mother. So that would suggest that there's no sex linkage of that genomic region, and that it was just part of the autosome. Another possibility is that maybe that particular position in the genome, that T was faithfully inherited from mother to daughter, which would suggest that rather than being autosomal, it was actually part of a sex chromosome. In this case, it could have been part of a W chromosome, which is found only in females. So the mother transmitted that T on her W chromosome to all of her daughters, which also have a W chromosome, and that caused them to develop into females. It doesn't mean that the T is a causative mutation that causes female development, but it's linked to the gene in this example that would lead to female development. And so we can do this by surveying variation in a mother and a father and many offspring and families across the entire genome. And we have at our disposal lots of data from reduced representation genome sequencing and also complete chromosome scale genome maps or genomes that we can use for mapping our data. And so what we can come up with are basically plots like this, where each of these boxes represents a chromosome. So there's 18 pairs of chromosomes in most of the Xenopus I'll be talking about today. And on the x-axis is just the chromosomal position. I know it's a small font here, but this would be chromosome 1L, and this is the beginning of the chromosome at a telomere, and that's the end. And then there's another chromosome that has a beginning and an end. And then we have on the y-axis the probability that a particular variant is associated with the sex of the offspring. So this is sort of like a GWAS study. It's basically a genome-wide association study that looks at the association between variation at the DNA level and a phenotype, which in this case is the phenotype, the sex phenotype of each offspring. And you can see, of course, that one part of the genome has a cluster of variants that are associated with sex, which is indicated with the hot colors. And so that in this particular species, which I'll show you in a moment, chromosome 4L appears to be the sex chromosome because it has genetic variation that's linked to the sex of offspring. And so we can do this across many species, and we've done this, and we're continuing to do it. And we've really uncovered what to me was striking and surprising variation among closely related species in the genomic locations of their sex determining regions. And so this is just a simplified depiction of some of the chromosomes that we've mapped in these different species. And you can see among these, within the genus, for example, there's at least 7 separate sex determining systems. And some of the same chromosomes have been co-opted multiple times independently to become sex determining regions or sex chromosomes in these different species. So there's a lot of variation in sex determining region in these frogs, which again underscores the phenomenon of developmental systems drift, wherein we're seeing, even though all of these frogs have separate male and female individuals, each of them that we've looked at and managed to map the sex chromosomes, almost every single one of them has a different sex determining region that is evolved independently. And more broadly, we've looked across diverse, sort of a broader phylogenetic scale, and we see the same pattern, maybe to a slightly lower degree, but still a really incredible diversity across other amphibians. And we can detect the variation among species here by looking at, in a phylogenetic context, about which sex is the heterogametic sex. So which sex has different gametes. So do they have an XY or a ZW sex determining system? And you can see in this just simple plot with only 100 species for which we have data, there's many turnover events where closely related species often have different sex determining systems, even at this broad phylogenetic scale. So developmental systems drift with sex determination is rampant in amphibians, and it's particularly so in Xenopus. So the next question I want to explore with you is how what we call the recombination landscape might influence developmental systems drift and influence where new sex determining genes arise. And so what do I mean by the recombination landscape? What I mean is where in the genome and how frequently recombination occurs. And I want to divide that up by sex and argue that recombination isn't a phenomenon that's the same in both males and females. It happens during gametogenesis or oogenesis and spermatogenesis, but it turns out in most species, including humans, it happens in different ways in oogenesis versus spermatogenesis. And so in many species, including humans and including the frogs I'll be talking about today, There are hot spots and cold spots of recombination in the genome, whereas some areas of the genome recombine a lot and other areas don't recombine very much at all. And as well, there's also different rates and locations of recombination, or the probability of recombination between the sexes. So in humans, for example, and also it turns out in frogs, the rate of recombination is higher in females than it is in males. And also the locations of recombination in females tend to be more towards the centromeres and less towards the telomeres, whereas in males, recombination happens more frequently in telomeres than it does in centromeres. And so, this means that this is what we mean by the recombination landscape. How does recombination vary across the genome, and how does it differ between the sexes, both in rate and in location? And so to explore this, you might ask, well, I guess we could begin with an observation. What we often see when we look at sex-determining regions is that they're frequently embedded in genomic regions that basically lack recombination. So this is true, for example, in humans, where the SRY gene is on the Y chromosome, and most of the Y chromosome does not recombine with the X chromosome. So it gets inherited as an almost complete chromosome from father to son. And so why might that be the case? And there's lots of hypotheses out there. It could be that recombination suppression is favored to resolve genomic conflict associated with different fitness values of each sex. It could be a way to ensure that a sex-determining gene is faithfully inherited from parent to offspring of the same sex. It could be that natural selection on regulation of or control of expression influences recombination suppression, or it could be a neutral phenomenon. And all of these explanations are not mutually exclusive, and possibly it may collectively contribute to recombination suppression around sex-determining genes. But another possibility is that genomic regions that harbor sex-determining genes may themselves have a pre-existing low rate of recombination, and therefore be more susceptible to receive sex-determining genes that evolve de novo, because that's sort of a suitable place that natural selection might favor a sex-determining gene to arise. And so this idea that there could be a chicken and egg situation here, that recombination suppression could come after a trigger for sex determination, or it could come before a trigger for sex determination arises, is, I think, an interesting one to explore. And it turns out that these frogs really allow us to do that very effectively. And so in order to explore this question, which came first, sex determination or recombination suppression, we need to first characterize the recombination landscape in the frogs that we're looking at. And we can take advantage of the same data that we use to map sex determining regions to do this. And so we begin with a chromosome scale genome assembly, and we also have a large family that we've raised, that we have individuals, parents, and also offspring of known sex. And then we have reduced representation genome sequences that allow us to basically build linkage maps for SNPs that are found, or single nucleotide polymorphisms that are found only in the mother, and then those that are found only in the father. And then that can allow us, by comparing to offspring, we can look at how those SNPs are associated or not associated, and infer how recombination happened during oogenesis in the mother and during spermatogenesis in the father. And we can compare the recombination events. We can ask how often they occur, and also where in the genome they occur in each sex. And so we've done that with 3 species so far, and we found really consistent results in these 3 species that actually are basically exactly what people have observed in humans, where if you look at this density map, this is across the whole genome. We've, on the x-axis, we've looked at the relative chromosome position. So there's different size chromosomes, but we can sort of scale them all to be the same size and look at how recombination occurs. And we see in general that in blue here, maternal recombination events tend to occur in the centers of chromosomes. And then in brown here, you can see that paternal recombination events tend to occur on the telomeres towards the end of chromosomes. And this is true in 3 separate species that we've looked at, suggesting that this is sort of the ancestral condition of the recombination landscape in these frogs. And so you can imagine that if it's beneficial for a trigger for sex determination to exist in a place that has low recombination, in our case, for Xenopus, the triggers for sex determination are female inherited, so they're on a W chromosome. So we'd expect them, based on this recombination landscape, to be situated on the tips of chromosomes. And that's because in mothers, the recombination is lower on the tips of chromosomes. That's where the blue density maps are low towards the ends, whereas in fathers, it's high. So because it's a maternally inherited female-specific trigger for sex determination, we would predict that if there's a pre-existing benefit to having low recombination, that these triggers for sex determination would be on the tips. And so we can actually map where these triggers for sex determination are and test that hypothesis with the genomic data we have. And so this is the chart I showed you earlier that shows the phylogeny of these different frogs and the locations of their chromosomes. And so let's look where on each of these sex chromosomes the sex-linked region actually is. Is it in the center, or is it on the tips as we'd expect? And so, for example, in this species, Xenopus melotropicalis, again, we've got the genomic position of chromosome 7 on the x-axis and the probability of association of single nucleotide variants on the y-axis. And you can see that there's a strong signal on the end of the chromosome and towards the beginning of the coordinates for this species. So that's consistent with our hypothesis. Above each map, I'm showing you a density of genes. And so in this case, there's a high density of genes in the area that happens to also harbor the sex-determining gene. We can look at other species and we see a pattern that very much echoes this finding in Xenopus melotropicalis. So for example, in Pygmaeus, we see on chromosome 8, there's a sex-linked region on one end of the chromosome. And then in Xenopus allophrageri, we see a weak signal of a sex-linked region on the beginning of chromosome 7, in the same place actually as melotropicalis, but we think it's independently evolved. And then we can look at other species as well. Laevis has its sex-determining region, again, on a tip of a chromosome, where recombination is low in females. Same with Borealis on chromosome 8. It's actually on the opposite side of the chromosome 8 that Pygmaeus has for its sex-determining region. And then sorry, that's this one here. And the same is true of Mulleri, which chromosome 4. And the only exception we see is in Xenopus fishburgi, where the sex-determining region is right in the middle of the chromosome, even though it does have a female sex-determining system. So, in general, we're seeing, indications that our predictions are borne out, where generally the sex-determining regions arise in areas of the genome where recombination in females is relatively infrequent or comparatively infrequent. And so we're in the process of developing permutation tests to formally test this and add some statistics to it. So the last topic I want to talk about is our work with gene editing. And it basically asks what sex-determining genes do now and in the past and present. And to do this, we're working with arguably a quote-unquote relatively simple system for sex determination. And so the only gene that's known to trigger sex determination in amphibians actually turns out to be from Xenopus, and it's a gene called DMW. And this is it's a female-specific gene, and it's the trigger for female sex determination in this frog, Xenopus laevis. So DMW, turns out, was formed from partial gene duplication of the male-related gene DMRT1, which I alluded to earlier. And so it's a paralog or a gene copy of a sex-related gene. And as we'll see, there's a relatively complicated genome here because Xenopus, most Xenopus species are polyploid, and Xenopus laevis in particular is a tetraploid species, which means it has 2 copies of most of the genes in the autosome, DMW being the exception. This is a gene that's on the sex chromosomes, and it's present only in 1 allele in females and 0 alleles in males. And so a large part of DMW was formed from gene duplication of DMRT1. That includes the 5' untranslated region and also a DNA binding domain called the DM domain. And so it's thought to actually, when translated, it probably binds to the same regulatory regions that DMRT1 does. So both of these are transcription factors, and they either turn on or turn off expression of male-related genes, we think. And so there's a sort of simple model for how sex determination is affected in Xenopus. And so it works like this. In males, we have DMRT1, which is an autosomal gene. It's a transcription factor, and it binds to the regulatory element of male-related genes and causes the male developmental pathway to ensue. And then in females, at the time of gonadal differentiation, DMW is expressed, and we know this is true from various studies that use qPCR. And we think that DMW binds to those male regulatory regions of male-related genes and prevents their activation by DMRT1. It sort of blocks male differentiation and causes the default pathway of females to ensue. And so that's the model that we have. Of course, it's more complicated than that because Xenopus laevis is tetraploid, and then there's also some other genes that are female specific. And so I want to talk about these genes. It's still fairly simple, but it's got more moving parts. And so in addition to DMW being found only in females, there's two other genes, ScanW and CCDC69W. So these are genetically linked to DMW, and they're found only in females in one allele. They're not found in males. And then DMRT1, because Xenopus laevis is polyploid or tetraploid, it has two alleles of DMRT1. Both of them have two genes. Both of them have two alleles. So we call one of the genes DMRT1L and another of those genes DMRT1S. And both of them, as we'll see, actually have different function, it turns out. And they're present in the autosomes. And so DMW, if it is to block the sort of male differentiation, it needs to block both of the homologs or paralogs of DMRT1 that are present in the autosome. And both of them have DM domains that appear functional. And so to explore this system, we've been using genome editing with a CRISPR-Cas9 system. And just briefly, I think many of you have probably heard of this. There's a protein that was isolated from bacteria that forms a ribonucleo catalytic complex with what's called a single-stranded guide RNA. And this single-stranded guide RNA has a recognition sequence that can target a particular portion of the genome for cutting. And when we inject this protein and the guide RNA into embryos, it can home in on a very specific region of the genome that we've specified by customizing the guide RNA sequence, and it causes cutting, which is then repaired through non-homologous end joining, which is an error-prone mechanism that frequently introduces deletions and insertions. And so if we target sort of the 5' region of the coding region of a gene, we can often introduce frameshift mutations that essentially makes a null mutation. So it inactivates a protein. And there's also ways to add in proteins as well. And so we've done genetic manipulations of these frogs by knocking out all of the female-specific genes, and as we'll see, each copy of DMRT1 as well. And so we can start by looking at our results with DMW. So we introduced a 10 base pair deletion right after the start codon, and so this causes a frameshift mutation and basically a knockout of DMW. And in these individuals, we get a complete female to male sex reversal. So they're genetically female, but they have an inactive version of DMW, and they develop into phenotypic males. So completely normal, fertile phenotypic males that amplex with other females. And so we can look at them at a cytological level. And so this is a wild type testes. You can see in dotted lines here is a structure called the seminiferous tubule, and this contains fertility cells and also late spermatids, which are kind of hard to see, but they're sort of the dark purple stain here. And we can look at these females that have testes because they've been sex reversed, and you can see that there's completely functional seminiferous tubules, and you can see the sperm inside these functional testes of these sex reversed females. And so we've demonstrated that these females can not only have mating behavior with wild type females, but also they are fertile. So we can make offspring that have that carry this DMW knockout gene into multiple generations later. And so that suggests that this is indeed the sex determining gene in this species, with some caveats I'll talk about in a moment. So what about ScanW and CCDC69W? What role did they play in female differentiation? And so to test that, we introduced deletions, frameshift mutations in the 5' region of the coding region of each of these genes. And you can see those mutations here. And again, these are frameshift mutations that cause basically a null protein of each of these each of these genes. And the phenotypes actually were none. So we didn't observe any discernible effect of knocking out SCAN or CCDC69W in females. So these females, the knockout lines were fertile. They developed into normal females. Everybody looked happy. And so what this demonstrates to us is that these two genes, although they are female specific, they're not necessary for female viability, fertility, or female differentiation. And so our findings from this part of the work suggest that DMW is a likely trigger for female differentiation. This is consistent with other reports by other groups that have suggested this as well. There are some caveats because there could be another female-specific gene that operates upstream of DMW that's not identified, but I think there's fairly low chance of that being the case. And then they also tell us that SCAN and CCDC69 are not necessary for female differentiation, and as well, they don't prevent male viability, differentiation, or fertility, because we know that females that have the knockout gene of DMW develop into males that are completely functional and fertile and viable. And then again, these two genes are not necessary for female viability or differentiation or fertility, because when we knock them out, females still develop into normal females. And so this tells us about how the functions of different female-specific genes vary, and vary not only in what they do, but also whether they are necessary for female viability, differentiation, or fertility. We see one gene is, but two of them that are still female-specific are not. So we wanted to explore this further by looking at how DMW impacts or interacts with DMRT1. And we did this by then going on to knock out DMRT1L and DMRT1S in Xenophus laevis. And so the CRISPR deletions that were introduced both happened to be 7 base pairs. They were in unique 7 base pair mutations that were in the coding regions of each gene and in separate lines. So we haven't made the double knockout yet for these genes. We were able to then use PCR to assess whether there's a sex reversal phenotype. So when we knock out DMRT1L or DMRT1S, we could tell what genetic sex it was by amplifying DMW. So if it's present, then it's a female genetically, and if it's absent, then it's a male. And then we also wanted to see what the effect of knocking out these genes are on the phenotypic development of each of these sexes. And so what we found was that neither knockout caused sex reversal. So here's another image of a wild-type testis. And you can see when we knock out DMRT1S, they still develop testes that are more or less normal. And these male individuals we know are fertile because we've managed to cross them to wild-type and produce normal offspring. For females, we also found that the individuals are female, and when we knock out this gene, and they're also normal and fertile. So it appears that DMRT1S is neither important or necessary for male differentiation nor for female differentiation. So its exact function we're not exactly sure of at this point, because we don't see we see a weak phenotype in testes, but it's not one that completely knocks out testis function, and we see no phenotype in females. DMRT1L, on the other hand, had a strong and severe phenotype in both sexes that was quite distinctive. So again, here's a wild-type individual, and you can see the seminiferous tubule and then normal sperm in maroon, in purple. And you can see here in this individual male, and we knew it was male because it didn't amplify DMW, but it did amplify a positive control. You can see that the seminiferous tubule is present, but the sperm cells are not, or at least they're aberrant. So you see dots here rather than the sickle shaped. And these individuals we know don't produce normal sperm. We've looked at it with a microscope, and also they appear to be sterile. We haven't been able to produce offspring even with in vitro fertilization. So that was one of the types of individuals we saw. And then another type of individual had no testes at all. And so I've only shown 2 here, but we found about half of our offspring that had homozygous DMRT1L knockout, if they were genetic males, did not develop testes at all. And strikingly, when we looked at females, we were surprised to see that all of the females did not develop ovaries at all. So there was completely absence of gonads in females. And we were really surprised to see this because in humans, for example, when DMRT1 is disabled, it doesn't prevent female differentiation. It only prevents male differentiation. And so, excuse me, this appears to be a novel function for DMRT1, at least this homeolog of DMRT1 in Xenopus laevis and potentially in amphibians in general. And so how can we interpret these functional differences between these two duplicate genes that presumably started off their lives as having the same function because they were complete duplicates of one another? And so there's sort of two main evolutionary hypotheses for how divergence in function happens after gene duplication. And one is called subfunctionalization. And the idea is that in the ancestor, you've got two distinct functions indicated here with a square and a circle. And then through degeneration of subfunctions, you end up with divergence in the descendants. So for example, in L, they could have lost the blue square function, and in S, it could have lost the red circle function. And so that leads to divergence in functions among homeologs. So there's more specificity of these homeologs because the ancestral gene was multifunctional, and then one or the other function was lost during evolution. And then another scenario is called neofunctionalization, where ancestrally there could have been one function, and then maybe one or the other paralog gained new function. So in this case, the blue function became a red circle function. And so to distinguish between subfunctionalization and neofunctionalization, the typical way that evolutionary biologists do this is to look at an outgroup species and ask whether that outgroup has a gene that's multifunctional in the case of subfunctionalization, or that has a single function in the case of neofunctionalization. And so we're able to do this by knocking out the gene DMRT1, which is present in a diploid species called Xenopus tropicalis. So this would be an autosomal gene in tropicalis with two alleles, but it doesn't have the two copies that you see in Xenopus laevis, because Xenopus laevis is a polyploid. And so we did this, and what we found is that when we knocked out DMRT1 and made a homozygous knockout in Xenopus tropicalis, the males were able to form testes and they were able to form sperm. So the sperm appeared to be lower in count, but it was still capable of making progeny. So males appear to be fertile when they completely lack DMRT1, which is a surprise for us. Females, interestingly, had a different phenotype where there's no ovaries and they were completely sterile, obviously, because they have no eggs. And so we see a sort of similar phenotype. It's actually DMRT1, not DMRT1L, for tropicalis, as we do in the DMRT1L knockout in laevis. And so that allows us to make an evolutionary interpretation of how function changed over time. So we see, for example, in females, that there's sort of evidence for subfunctionalization in the sense that the DMRT1 gene appears to be necessary or essential for ovarian development, but that function was lost in DMRT1S and Xenopus laevis after gene duplication formed that gene. So DMRT1S is dispensable for female differentiation. In males, on the other hand, it appears that when you knock out DMRT1, you still have normal testes in tropicalis, but when you knock out DMRT1L, you actually have abnormal or no testes in Xenopus laevis, which suggests that this gene has become newly functioned or neofunctionalized during the evolution of DMRT1L in Xenopus laevis. And so this gives us sort of a sex-specific perspective on functional evolution of DMRT1, where there's evidence for subfunctionalization in females, but neofunctionalization in males. What's interesting as well, and this is what I'll sort of end with an anecdote, is that we can see in both sexes that this one homeolog, DMRT1S, is not essential in females and males. You can knock out DMRT1S in females, they still are fertile. You can knock out DMRT1S in males and they're still fertile. So it appears that DMRT1S is not essential and not less important than DMRT1L. So that's relevant because I haven't told you the whole story about how DMW arose. So I said that it arose through partial duplication of DMRT1, but it turns out that we know more than that. So we can look at the species tree of Xenopus laevis. So we have a diploid outgroup. There were two diploid ancestors that I've got depicted here with red and blue. They underwent a process called allopolyploidization, where their complete diploid genomes were fused into a tetraploid ancestor that gave rise to Xenopus laevis. And so that's why we have these two genes, Xenopus laevis, DMRT1L and S. It turns out that DMW arose after genome duplication, and arose through copying DMRT1S, not DMRT1L. And we know that definitively based on a phylogenetic analysis of the sequence of all of these genes. And so what this suggests is that this pivotal sex-determining gene in Xenopus laevis arose through gene duplication of a marginalized copy of DMRT1, DMRT1S. So what this argues, I think, is that DMW is derived from this ancestral gene that may have been non-essential. And this suggests that there may be, an important role for what we call tipping points in biological phenomena and in developmental systems drift, where small changes in established genetic networks can have huge consequences. And we see evidence for tipping points in the climate, we see evidence for tipping points in road rage, and we see it again in developmental systems drift in biology. So small changes can have big consequences when you're at the margin of a decision junction in biology. And so future work that we're doing now is to go on and use comparative approaches to better understand exactly when and what the function of the ancestral gene of DMW was. And so we're going to take advantage of another tetraploid species called Xenopus borealis. And so we can look, this is a species that's sister or closely related to Xenopus laevis, and it also is allotetraploid. We can sort of pull this species tree apart and get a gene tree that has an L lineage and an S lineage. And we know that DMW arose before the speciation of Xenopus laevis and borealis. And so by looking at knockout phenotypes of borealis, DMRT1S and DMRT1L, we can get a handle on whether it also has sort of non-essentiality of DMRT1S, which again would support this hypothesis that DMW arose from a non-essential ancestor. And so that's a work in progress that we're very much enthusiastic about pursuing. So to summarize, what I've talked about today suggests that there's a lot of variation among species in Xenopus and sex chromosomes, which suggests that there's many different sex-determining genes that are scattered throughout the genome in these different species. There's sort of many careers of work left to do on the system. We see possible influences of the recombination landscape, where we know that recombination is less frequent in females on the tips of chromosomes, and that often is the location where we see sex-determining regions arise, suggesting that natural selection may favor the origin of sex-determining genes in parts of the genome that already have low recombination rates. And then finally, we see evidence through our knockout experiments of dynamic functional evolution that highlights the importance of tipping points in biological innovation, where we see the origin of a key trigger for sex determination arising from an ancestral gene that probably was non-essential in the ancestor when that duplication event happened. I also just want to talk more broadly very quickly about some of the exciting aspects of amphibians for understanding sex chromosomes. And so, this is a group where we have a lot of evolutionary replication. So there's a lot of examples of new sex determining systems that have evolved. There's a lot of species diversity. There are genomic tools like chromosome scale genome assemblies. We have large clutch sizes, so we can generate families and actually document a lot of recombination events, which can give us statistical power to infer where and when and how frequently recombination occurs. And we also now, I think most excitingly, have the ability to manipulate the genomes with genetic engineering by doing microinjections, which is depicted here, where these relatively large zygotes can be injected with genome-altering substances that can allow us to study the effect of individual genes. So this can allow us to ask questions such as what types of genes become sex-determining genes, how do they arise, and how do they function? What other genes are paired with them? So we saw a scan in CCD-C69 along with DMW. What are they doing in the genome? And also, ultimately, what drives sex chromosomes turnover? Why do we see so much variation in some groups but not in others? I would say as well, I haven't talked about it extensively, but amphibians are also exciting because there's lots of instances of polyploidization, especially in Xenopus, which allows us to explore how that may catalyze biological novelty and change in genomes. And also there's lots of examples of gene flow among species, which allows us, yet another dimension of scrutiny for how novelty can arise in different species. And I'd say finally, and I mentioned this when I was talking about the acknowledgements, this has been a really rewarding aspect of my work, that I've had the opportunity to work with collaborators in other countries where amphibian diversity is very high. So we work mostly in sub-Saharan Africa, where Xenopus is native. And I've been able to work with just amazing collaborators there that allow for, bidirectional exchange of ideas and scientific productivity. So yeah, thank you very much for listening. I hope you enjoyed it, and I would be very happy to answer any questions Matthias may have, or if anybody would like to email me, they can do that as well. So thanks.
Thanks, Ben. This was great. And what I'm enjoying about recording this series is that each and every episode really illustrates how multifaceted it is. Anytime, anyone wants to study evolution, you can approach, evolution in so many different aspects. And I'm really enjoying that diversity of approaches. Now, I do have a few questions for you because, this topic was so foreign to me. And I was like, wow, it's something that I didn't think about. And it's very interesting. And so the first question that I have for you is, how often do we see sex-specific neo-functionalization or sub-functionalization in other genes? Do we know that? Do people study that?
My understanding is that there's very little information on it. So sub-functionalization and neo-functionalization can both happen. So we can get degradation and novelty in the same set of genes. But I'm not aware, and that's not to say there isn't an example, but I'm not aware of sex-specific examples of either of these. I think it happens a lot, but I think it hasn't been documented very well yet.
Interesting. And specifically talking about, if I got the name right, so we have one DMRT1S paralog, right, which is apparently not important right now. So in this particular case, so for genes that we cannot find functions, do we think, I know it's hard to come up with generalizations, but do you think it's a case in which the gene itself is really not important anymore, it's just going to drift away and become a pseudogene, or we just haven't really found a phenotype for that gene? We haven't really come up with the right experimental design.
Yeah, I think it's probably, that's a great question. I think it's probably the latter. I think that, so we do see a minor phenotype in the knockout males where the sperm looks like it's lower count and sometimes not fully developed, but they are fertile. We know that from other people's work that there is differential expression of DMRT1 L and S. So the L is expressed in the germ cells and the S are expressed in the supporting somatic cells in the gonad. And so there probably is an important or relatively important function that we haven't been able to get to actually assay with the appropriate experimental design. So I think you're right. It's probably not just doing nothing. It's probably doing something that we haven't really been able to fully elucidate. So we have done RNA-seq for all of the different knockout genes that I've talked about. And we do see transcriptomic effects of all of our knockouts. And so that hasn't translated to our coarse phenotypes of viability, fertility, and differentiation, but it does have, a molecular consequence for every one of the genes, including SCAN and CCDC. So yeah, it's a great point. All of these genes, if they're around in the genome, they're probably doing something. And you could imagine that, in the right context, you could see a fitness difference as well, but we just haven't observed it.
Agreed. And now, I did understand, when you were explaining the chicken egg problem, in which where the sex determining genes, where they are lying in the genome, right? And those regions, they often do not recombine, or they recombine much less. And I understand it's hard for us to tell whether those genes were already in those regions, or because they migrated to those regions then stopped recombining. But I was really curious, do we have any theory as to why we have sex-specific recombination landscape differences?
Yeah, so that's another area that's very interesting. So there's several hypotheses out there. One hypothesis is that it's just neutral variation, that there could just be drift in recombination rates between the sexes, and there's no fitness benefit to it. Another hypothesis is that it's something to do with sexual antagonism, that it's a way to resolve genomic conflict. It could be something to do with the fundamental differences between oogenesis and spermatogenesis. I mean, they're really massively different. Like spermatogenesis, every event produces 4 gametes, 4 sperm. Oogenesis produces 1 and then polar bodies.
Yeah.
And, it doesn't even go to completion in humans. It gets arrested at, at different stages, depending on, before ovulation, and then even after ovulation, it gets arrested before the final polar body gets left out. So they're really different processes that I think are probably poorly represented in a lot of textbooks, how different they are. And so it could be that there's something about the mechanics associated with these different biological processes that causes recombination to happen in one type of gametogenesis more than another and in different places. But yeah, I mean, I guess the short answer is we don't have a good explanation of exactly why we see these differences in the first place. And it's a really exciting question that I wish we knew more about.
Yeah, absolutely. Again, as I said, all these things, I really actually did not know any of this. So I was very astonished. And one way that as well, to wrap things up, I really liked when you brought the concept of tipping points to the developmental systems drift. And as you mentioned, tipping points are usually we relate them to climate studies, right? And I think it's becoming more and more clear to us that you can find those parallels at the molecular level. Just yesterday, I was reading about this one researcher who now coined the term the keystone gene. So this guy studied how one particular gene in an artificial food web system, in a small food web system, the different alleles really impacted the composition of the food web. So he coined the keystone gene, which is analogous to keystone species. So yeah, I think evolution at the molecular level, it's funny to say it's almost like it's still in uncharted territory, right? And this is what the series hopefully will showcase to students and people who are interested in the field.
Yeah,
Ben,
I mean, I think that's absolutely true. Yeah.
well, Ben, this was great. Thank you very much. Again, I'll stop the recording now, and then the recording will be posted on YouTube and our website. And we can chat for a few minutes as well. Let me just stop. So thank you again, Ben, for being here with me
Yeah,
today.
thank you for having me.